Okay, I want to start by thanking the organizers of this seminar series for extending an invitation to me to present some of the results in my lab on nano-delivery of novel inhibitors of DNA repair for enhanced cancer therapy. I'm hoping to be able to make a case for the use of nanomedicine in the drug development process by the end of this lecture. So at the beginning, I just wanted to disclose some conflict of interest, mentioning the polymer-based nanoformulations of Pnkp inhibitors. They are covered by these patents, and also the functionalized caprolactone monomer and other functionalized polylactones used in the research are also covered by worldwide patents as listed here. And also wanted to mention that the functionalized caprolactone monomer and also the polymer, they are licensed to Meros polymers. Since the introduction of Doxil to the market in 1995, the growth in the use of nanomedicine has witnessed a steady pace. And today there are over 100 nanomedicine formulations approved by FDA since then. This picture only captures some of these well-known nano products that are used in the pharmaceutical industry, but there are way more materials that are approved, as I mentioned. Most of these products target oncology areas, around 35%, but there are other areas where nano delivery system can also play a role and induce some benefits. So in terms of the global market for nanomedicine in 2009 was around $53 billion. And this has grown tremendously, at least 3 times over the years, and then maybe 5 times around 2017, and today around 5 times today. And this is not even considering, I guess, the COVID vaccines that has been, products of nanomedicine, some of them. So in these cases, most commonly, nanomedicine is either used to improve the performance of current therapeutics in one of these forms. For example, increasing the solubility of some small molecules, or increasing their biological half-life, reducing drug distribution to healthy tissues, and by doing that, reducing the side effect of the drugs and increasing the quality of life in patients, or enhancing drug distribution and access to the diseased tissues, cells, and intracellular organelles and targets. So these are some of the areas that the current nanomedicine has been used on. Other areas was to develop new medicine from challenging molecules, molecules that have difficulty in their delivery. For example, large molecular weight materials such as protein drugs or gene therapeutics, or for the development of vaccines. So these are the areas that the nanomedicine has been covering so far. One area that has not been looked at maybe enough is the area of drug development for small molecules. So we all know that the drug development is a lengthy process, and it is a very inefficient process. So from every 10,000 compounds that are made in different laboratories, only maybe one gets into the market and into clinic. And it is a very expensive process as well. So nanomedicine might be able to help make this process more efficient and also expedite the pace towards drug development. But perhaps to be able to convince the pharmaceutical industry to invest in nanomedicine in the drug development process, we have to find a good niche. I mean, find an area where, for example, targeted drug delivery is really important, where nanomedicine can really be a game changer. So in my laboratory, we have been working on this niche for a long time, especially focusing on drugs that can sensitize cancer cells to conventional chemotherapeutic agents. And recently, we have been working on inhibitors of DNA repair in this regard. So we think that nanomedicine can offer several advantages in the delivery of inhibitors of DNA repair for cancer therapy. A lot of anti-cancer agents and also radiation therapy, they cause harm to cancer cells because they damage their DNA. And the capacity of the cancer cells to repair this damage can make cancer cells resistant to DNA damaging therapeutics. Now, DNA repair inhibitors can reverse this resistance and make cancer cells again sensitive to chemotherapy or radiation therapy. But for at least two reasons, it is very important that the DNA repair inhibitors be targeted to cancer cells and only to the tumor. The first reason is that if they are going to be used in combination with systemic chemotherapy, you don't want to make normal cells sensitive to the effect of DNA damaging therapeutics. And the second reason is that the DNA repair inhibitors over long term of use, because they cause genetic instability in normal cells, they may eventually in long-term use may cause cancer themselves. So therefore it is important to target DNA repair inhibitors to cancer cells. DNA repair inhibitors found their way to clinic in 2014 with the approval of PARP inhibitors. Olaparib was the first drug that has been approved, and it is available as an oral medication. In this case, again, because targeting cancer was important, what has been done was to target cancer cells by a process known as synthetic lethality. So what is synthetic lethality? It means that some cancer cells, they might be deficient in some proteins or some genes that makes them especially susceptible to the inhibition of, for example, PARP. In case of PARP, for example, we know that PARP is a protein that is involved in single strand break. Now, if DNA is damaged by single strand break, now PARP can repair that damaged DNA, but eventually this DNA can again be repaired by BRCA. Now, if BRCA is mutated in cancer cells, now this cancer cell will become specifically more susceptible to the inhibition of PARP. So inhibition or mutation of BRCA and inhibition of PARP, it causes a synthetic lethal partnership between these two proteins, and as a result of this synthetic lethality, but the cancer cannot survive and it will die. So that is basically the mechanism by which PARP inhibitors have found some sort of cancer targeting effect, and that is why that PARP inhibitors are all approved for use in patients with BRCA mutated genes. Now, at the University of Alberta, I have been lucky to get acquainted with Dr. Michael Weinfeld, who has been working on a different DNA repair enzyme for a long time. And we have teamed up with another scientist and chemist, Dr. Dennis Hall, to develop new inhibitors of this enzyme, which I abbreviate as Pnkp. So Pnkp stands for polynucleotide kinase phosphatase. It is an enzyme that has both kinase and phosphatase activity. Now, if DNA gets damaged by the function of ionizing radiation, reactive oxygen species, or topoisomerase inhibitors, it can basically form this kind of unusual terminus where the 3' end of the DNA is phosphorylated and the 5' end is hydroxylated. Now, Pnkp is an enzyme that can work on this specific kind of damaged DNA and reverse this basically unusual termini of the DNA, make the 3' again hydroxylated and the 5' phosphorylated because it has both kinase and phosphatase activity. And now when the DNA is formed to its usual form, now it can be rejoined by the action of DNA ligases. Now, if Pnkp is inhibited, now we can think that the cells can become more sensitive to the effect of ionizing radiation and also topoisomerase 1 inhibitors. The research group of Dr. Weinfeld also have found a synthetic lethal partnership between inhibition of Pnkp and PTEN deficiency in cancer cells. PTEN is the second most common tumor suppressor protein after P53. Therefore, in normal cells or some cancer cells that express PTEN, they are not going to be sensitive to Pnkp depletion, so they will survive after Pnkp inhibition or depletion. But cancer cells that are deficient in PTEN, and those are actually more aggressive form of cancers, they will become sensitive to Pnkp inhibition, and through the process of synthetic lethality, upon Pnkp depletion, they will go through cell death. They have also found another tumor suppressor, SHIP1, to be a synthetic lethal partner of Pnkp, but SHIP1 is more relevant to hematological cancers, while PTEN is more relevant to solid tumors. So we made a hypothesis for the project that if we can find a nano-delivery system for a hit Pnkp inhibitor that was developed in our lab, and we can target the tumors with this nano-delivery system, then we can generate a specific cancer monotherapeutic for aggressive solid tumors that are deficient in PTEN. And also nano-delivery of a hit Pnkp inhibitor, they can specifically sensitize cancer cells and not normal cells to ionizing radiation and topoisomerase 1 inhibitors. As our hit Pnkp inhibitor, we have chosen this drug or drug candidate, I have to say, which was called A83B4C63. This was developed in the laboratory of Dr. Dennis Hall, as I mentioned, and these are the specifications of this compound for Pnkp inhibition. So as you can see, it has a very low water solubility. This has to be micromolar, and then the log D is relatively high. Because of the high log D and hydrophobicity of this compound, as the nano-delivery system, we chose to use polymeric micelles. So just as a reminder, polymeric micelles are made from self-assembly of amphiphilic glycopolymers. Upon self-assembly of the amphiphilic glycopolymers, they form a core shell structure, And then this core can be solidified depending on the structure of the polymer that makes the core. And the nano delivery system is somewhere around 100 nanometer or less. Now, as the building block for the polymer, we chose to use either polyethylene oxide polycaprolactone or some derivatives of polyethylene oxide polycaprolactone. This polymer has been generated first by one of my first PhD students, Dr. Abdullah Mahmoud, who is currently working in the FDA. So as I mentioned, we either use the original POPCl, which is a known polymer. Here, our group is hydrogen, or we use some functional groups instead of the R. The advantage of the new polymers that we have developed over POPCl is that, first of all, because this is made up of a backbone that is polyester, and we know that polyesters are used in absorbable sutures, so it can be considered to be safe. It has a potential for safe application in human. And another advantage that it has over PCl is that because of the substitution on the PCl, now the polymer will become amorphous. Therefore, it can be lyophilized and reconstituted after lyophilization. This is not achievable by the original POPCl, and that is a good advantage pharmaceutically. In addition, we can substitute different R groups here, and this will provide a chemical flexibility for the polymer. And as a result of that, we can match the structure of the polymer with the drug that we want to encapsulate. And therefore, we can basically have a solution for any drug with any physicochemical properties just by changing the R. Over the years, we have developed several polymers with different substitutions on the polycaprolactone. Some of them are listed here. We have attached fatty acids, we have attached cholesterol, we have attached functional spacers with thiol or hydroxyl functional groups. We have also carboxyl group, and we have been able also to chemically conjugate some drugs to this polymer. On this end, we have been able to basically attach peptides, monoclonal antibodies, and other ligands for targeting of cancer cells as well. In this specific project, we have used this polymer, the one with the benzyl carboxylate, at the core or the R here, at the core of the micelle or the R substitution. So this polymer platform has been developed in my lab, and this is the platform that has been licensed to Meros Polymers. So this work has been done by Zahra Shayer, who is currently a postdoctoral fellow, I think, in BC Children's Hospital. And she has developed the formulation of A83B4C63 in POPCl micelles and POPCl micelles both. So this is the size and TM picture of the POPCl micelles that she achieved. We looked at the release profile, and as you can see, the release was much slower when we use POPCl compared to the POPCl. And we then looked at the anti-cancer activity in form of colony-forming assay in cells, HCT116, which are the colorectal cancer cells that were wild type or PTEN positive, or those that were PTEN negative. And we saw anti-cancer activity only in the cells that were PTEN negative. Later on, my other PhD student, Sadat, actually continued that work and looked at the activity of this nanoformulation in vivo. So our studies in the HCT116 model, subcutaneous xenograft model, and that were PTEN deficient, showed that actually you can see that this picture speaks for itself. So this is how the tumor was grown for the control mice that received dextrose. This is the free drug that was dissolved with the aid of chromophore-EL and ethanol. And this is the drug in our nanoparticle formulation. So these graphs, the first one, the top one shows the caliper measurement of the tumor growth, and the second one shows basically the luminescence, the intensity of the luminescence from the tumor. And both data confirm that only the nanoparticle formulation of this heat Pnkp inhibitor was able to slow down the tumor growth. And the drug that was dissolved with chromophore-EL and ethanol formulation was not able to do that. We also looked at some other markers in the tumor. For example, we looked at the expression of Ki67, which is the biomarker for proliferation. And this also confirmed our previous result that only in the P10 negative tumors and only the nanoparticle formulation of A83 was able to reduce proliferation. But the drug that was dissolved with chromophore-EL was not able to do that. We also looked at the level of the drug in both P10 negative and P10 positive tumor models to make sure that there was no difference in the concentration of the drug because of the tumor model, and rule out any impact by the concentration of the drug. Basically, this result showed that our drug was only effective as nanoparticle formulation and only in the P10 negative tumor model, so showing synthetic lethality. We also looked at the biodistribution of the nanocarrier in the tumor and also major organs to find basically a reason for activity of the nanoparticle formulation. If you remember in the previous graph, this was the control. Empty nanoparticles didn't do anything. So it was not because of the nanoparticle alone or empty nanoparticles that we saw anti-cancer activity. So to look for the reason, we looked at the drug concentration. So at 24 hours, we have seen a rise in the concentration of the drug in the tumor, also in the plasma and in the liver. But at 48 hours, that was interesting that at 48 hours, almost the drug got washed off all the normal tissues except for the liver. And then we have seen a drastic increase in the level of the drug in the tumor. So if I put these two pictures together, it looks like that for the nanoparticle formulation, we see like a delayed accumulation of the drug in the tumor. And then by 48 hours, the drug is being washed off the normal tissues. And that is really important, especially if we want to combine this drug with chemotherapy, because we can start chemo after the drug is washed off the normal tissues. And by that, make sure that the drug is only effective in the cancer. So in the second part of this project, and to continue after synthetic lethality, we wanted to look at the effect in combination with chemo and radiation therapy. So we did some in vitro testing. Again, we used HCT 1.6 model, combined the nanoparticle formulation of A83 as free drug or nanoparticle with ionizing radiation. We have seen that in vitro, both free drug and the nanoparticle formulations were effective in sensitizing the cells to ionizing radiation. Similar effect was seen for in combination with irinotecan, which is a topoisomerase 1 inhibitor. We also did the same study in cells that were that had been knocked down of Pnkp through use of siRNA for Pnkp. So this is siRNA for Pnkp treatment. So we see the effect of Pnkp knockdown on sensitization to ionizing radiation. But when we give the drug to these cells, we don't see any additional effect. So in a way, this shows that our drug is specific for Pnkp. And then the same observation was made when we combined it with irinotecan. Now, recently, again, Sadat, with the help of Dr. Melinda Wust, he combined radiation therapy and Pnkp inhibitor in a mouse model. So for this study, we used a SARP instrument that can target the tumor with the radiation, with the beam of radiation. So these are our results, as you can see. Again, similar to the effect that we have seen with monotherapy in PTEN deficient model, we only saw effect for the nanoparticles in combination with ionizing radiation. So, and this effect was statistically significant compared to the control mice that only received the nanoparticle. I have to say that this study was done in HCT 116 wild type model, so the ones that were PTEN positive. So those became sensitive when combined with radiation. We also looked at the proliferation of tumor using FLT PET imaging in live animals. And again, similar result was achieved when compared to the caliper measurement of the tumor. We only saw a significant reduction in the proliferation of the cells in the tumor site by the nanoparticles of A83 combined with radiation compared to the control mice. And a third confirmation was achieved by looking at the same tumors under IVIS imaging, looking at the luminescence of the tumor, comparing the tumor size at the beginning of the treatment, at the end of the treatment. And as you can see, only for the mice that received the nanoparticles plus the radiation, we did not really see any significant growth of the tumor. And the other groups, they have shown growth when we compare tumors at the beginning and at the end of the study. We, imagining the medicine of the future. So we tried with collaboration with Dr. Wust and my postdoc, Igor. At the time, he was a graduate student when he did the study. So we tried to make these nanoparticles traceable through attachment of PET contrast agents. So we attached carbon-64 to nanoparticles that were actually modified on their surface with G11, which is a peptide that targets EGFR. So EGFR is expressed by many cancers, including some of the colorectal cancer. So we made this micelles. So we had two micelles. One, they were decorated with G11, and the other one, as a control, they were decorated with a mock peptide that used, a random sequence of G11. And then we were able actually to trace the nanoparticles in live mice and look at the distribution of the nanoparticles in live mice. So our strategy for the future is that if we can track the nanoparticle, we can see when it gets to the tumor and when it has the maximum accumulation in the tumor, then we can start either exposing the tumor with the radiation or start the combination with the chemotherapy. Again, just to, in further developments, We know that SN38 is the active metabolite of irinotecan. So Sadat has actually developed a nanoparticle formulation of SN38. And we have combined the SN38 formulation with A83. But I cannot disclose those results because we have to go for IP protection. But I just have to tell you that we found very interesting results when we combined the two nanoparticles in one nano-delivery system. Now, till this stage, we have been working mostly with this polymer PO-PBCl that have been developed in my lab. But one of the limitations here for further development is that, okay, PO-PBCl is, a new polymer, and then we are using a new drug. So it will become difficult and, challenging to bring this into clinic because we are dealing with two new components. So we tried then, using the whole library of the Pnkp inhibitors that we have developed to see if we can come up with another lipid or polymer-based formulation for some of these Pnkp inhibitors. So in my lab, so that's when the NMIN project started. In my lab, we have been working on some FDA-approved polymers. For example, PO-PCl. Here we used a longer chain of PCl compared to the one that we have used originally. And we have used PO-PDLA and another PO-PDLA. Like this last one is the component of Genoxo PM, which is a paclitaxel formulation in the market. And these are the results that we have got. Again, we use the same drug candidate, A83B4C63. They released a study this time, actually, when we use longer chains for these FDA-approved polymers, showed all of them could actually lower the release. Among these, the PO-PBCl and PO-PCl had the higher encapsulation efficiency and loading. But PO-PDLAs, they didn't encapsulate the drug that much. And also their size was a little bit larger than the other two. So based on this data, we decided to go ahead with the PO-PCl formulation and did a biodistribution study in the mice that with head and neck tumor, especially for combination with radiation therapy. Perhaps head and neck cancer was a better model than the colorectal cancer. So this is the size and the TM picture of the PO-PCl formulations that we have developed. Then we looked at the distribution and pharmacokinetic profile in mice. Unfortunately, we didn't see the same exciting distribution profile that we have seen for PO-PBCl with the PO-PCl. And these are basically the summary of the distribution profile in the mice. In collaboration with the Nanopore facility, we also chose other hit Pnkp inhibitors in the A83 series. So those were like molecules that could be loaded into liposomes using similar profile or similar formulation strategy as doxy formulation. Again, I just show you the basically slides for the methods that have been used by the Nanopore facility, and I cannot disclose data on these formulations that have been developed. And the reason is, again, because there is an opportunity for IP protection for the liposomal formulation. So basically use the same concept like gradients or remote loading process. And the drug, free drug that was not encapsulated was purified by ultracentrifugation, and that's what we have gone so far. So I wanted to summarize what we have found so far in this project is that, first of all, we have found that the nanoparticles of A83, B4C63 as our hit Pnkp inhibitor, They were able to inhibit the growth of P10 negative tumors as monotherapy, but A83B463 solubilized with the aid of conventional solubilizing agents was not able to do that. So if this drug or if this candidate was going through the drug development process with the chromophore EL formulation, it would have been eliminated. But now we see that as a nano formulation, it can actually be very effective, even as monotherapy. The nanoparticles of A83, they also acted as radiosensitizers in the wild type P10 or P10 positive colorectal cancer models. But again, the A83 solubilized with conventional solubilizing agents was not able to do that. And we have some promising nano formulations developed from Pnkp inhibitors using FDA-approved excipients. What we are going to do in future is that we are going to do a head-to-head comparison between the anti-cancer activity of the liposomal versus polymer-based formulations for some of these Pnkp inhibitors. The good thing is that both drugs that we have, either in liposome or in polymers, they are from the A83 series, so they are very structurally similar with minor differences that makes them suitable for either liposome or polymer-based delivery systems. Then we are going to use the best formulation we find here and use it in combination with radio and chemotherapy. I also wanted to mention that at the same time, we are working in my lab in collaboration with Dr. Weinfeld and also Dr. Fred West on other inhibitors of another DNA repair enzyme, ERCC1-XPF. This one can synergize with platinum-based chemotherapeutics and ionizing radiation, as well as cyclophosphamide. I wanted to bring to your attention that PARP inhibitors that have hit the market as the first DNA repair inhibitors. So they have actually, the global market was around like 900 million in 2018, and it was projected to grow by 27%, partly because of the growth in the incidence of cancers that were susceptible to these PARP inhibitors. And there are lots of different and big pharma playing a key role in development of these new inhibitors of Pnkp, inhibitors of DNA repair. So inhibition of Pnkp or new inhibitors of DNA repair can be projected to have at least a similar market. But with the nanomedicine, we may get a benefit for targeting cancer and being able to use it in a larger spectrum of cancer in combination with chemo and radiation therapy. So the market might be larger than this. And I hope that I was able to at least make you think about this potential for the use of nanomedicine in the drug development process. If nanomedicine can hit that niche and that market, there will be a boost in the use of nano-delivery systems in pharmaceutical products. And with that, I wanted to acknowledge different members of my group, current members and also the past members. Like those who are highlighted, they have played a role in the results that I have presented today. And these are some pictures from good old times when we could go to lunch and dinners together. And hopefully we will resume that soon. And I also wanted to acknowledge my collaborators, especially Dr. Michael Weinfeld, Dr. Dennis Hall, and Dr. Wust, for participating in this project, and of course, the people from the NanoCore facility at NMIN. And I'll be happy to answer questions.
So I'd like to thank you very much for that, for your talk. It's really very interesting. I'm opening the floor now to questions. And so while people are preparing their questions, I'd just like to ask a couple of questions of my own. One was and I didn't quite understand how this worked, but this is for for your polymeric nanoparticle. And you were saying that at 48 hours, you're getting much more delivery to the tumor than you were at 24. It looked like quite a dramatic effect. And I was just wondering what was causing that?
We are not sure. That's actually a really great question. And I have been attending actually the CSPS conference and actually gave the same talk there, and like with some modifications, but there was actually another presentation that they have used POPDLA, and they have seen similar, delayed delivery. And as you can, if you appreciate like the biodistribution, it doesn't seem that it is accumulating in any of the normal tissues and it is being released from any of the normal tissues. But that is something that we have to look into, and I'm really actually curious to see what is happening there. I'm not sure.
I see, because it was a bit unusual because you were getting more accumulation in the tumor as time went on, but less accumulation in some of the other tissues.
Yes, which is fortunate, I guess, for our purpose. Yes. But where do they go, and like how this happens, I'm not really sure. And I was thinking like maybe we have to look at I don't know, lymphatic system or somewhere, but yeah, you have to look into it.
Yeah, right. No, it looks like a very interesting, interesting observation. Okay, Marshall, do we have other questions?
Yes, we have a couple of questions that come in. Riyad Al-Srani actually asked the same question as you about high biodistribution of the drug in the tumor after 48 hours. So that's been answered, but just to acknowledge that question had been submitted. Himanshu Bhatt says, I would like to know about physical stability of these nanoparticles.
Yes, these nanoparticles, like the one with the benzyl group in the core, they are very stable, and that could be another advantage. And the reason for that is because of we think because of the pi-pi stacking of the benzyl group in the core of the nanoparticles. But we have looked at their stability before in vitro and in vivo. There's a paper that is published in Colloids and Surfaces with Shyam Garg as the first author. You can look that up. That entails the physical stability in vitro, that they are much more stable than the POPCL ones. And we have also looked at their in vivo stability in a breast cancer model, in a mice that bears breast cancer model. And that one is also published in Biomaterials with the same first author, Shyam Garg. So you can look that up, and yes, they are pretty much very stable.
Very good, thank you. Mohammed Bia has two questions for you. He wrote, thanks for the excellent talk. Then his first question is, what are the off-target effects of similar approved drugs compared to the nano-delivery system? And if you want to answer that, I'll read the second question out.
Sure. For Pnkp inhibition, as like approved drugs, you mean for the PARP inhibitors, or because Pnkp inhibitors are not approved. But for the Pnkp inhibitors, they are actually projected to have some side effects in the brain, in CNS system. That those studies are done actually in mice that were depleted with Pnkp. So they are projected to have some CNS side effects, but The good thing about the nano delivery systems is again that it will reduce actually their distribution to the CNS. For PARP inhibitors, I don't really remember what's their side effects. I have to look that up. Sorry about that. But you know, if they are combined with chemo, especially since they are given orally, you think that they may also make the normal cells sensitive to the effect of DNA damage. And in the long term, as I mentioned, DNA can be damaged in our normal cells by, you know, environmental factors, sunlight and things like that. And if you inhibit the DNA repair over time and it accumulates, it can lead to, you know, cancer and like genetic instability, as I mentioned.
Good, thank you. Mohammed B, a second question is, for colon cancer, can this nano delivery system be administered orally?
Orally, it doesn't, don't get the benefit. I'm not sure if it, you don't get the benefit of targeting. I know that you are thinking about, you know, maybe it gets into the cancer regionally, but it's very difficult to predict that, you know. For oral administration, and usually, you know, nano delivery system, they can enhance the solubility of drugs and enhance their bioavailability that way. But like it is very difficult to get the nano delivery system intact, getting absorbed from the GI tract, so you cannot benefit from the targeting. And we don't have any evidence that orally it can actually penetrate into the cancer. But this is something that, you know, we can perhaps look into. One thing I wanted to mention that from this polymer platform that we have developed, actually, we also have a thermoresponsive gel that, you know, that material could be applicable for local administration at, for example, the site of the colorectal cancer, with some form of, you know, similar devices that maybe they are used for colonoscopy or things like that. But this is something else that we have to look into.
Very good, thank you. There are no other questions at present, although Himanshu Bhatt says he would like to see the biodistribution slide again.
I can show that.
I have another question, Afsaneh.
Sure.
And that concerns where you say you're going to do a comparison of the liposome approach and the polymeric micelle approach. So just exactly which drug is it that you're comparing there? I think it is a weak-based drug, and so you can load it into the liposomes using the, as you outlined, the ammonium sulfate approach.
Yes, it is in this. It is a derivative of A83B4C63 that we have used in our polymer based material, but it has some functional groups that can get ionized. So it is not the same drug, because that same drug is not actually appropriate for loading into liposomes, but it's a derivative of that drug.
Right.
That it has actually similar activity to A83B4 in terms of Pnkp inhibition. Like it has similar activity, but its log D is lower because of that those functional groups. Yeah.
Right.
I did not disclose the structure because there might be an opportunity for IP protection for lipid-based formulations. I'm not sure. We have to look at the patents that we have right now and then see if we only covered polymer-based or maybe lipid-based can also be protected further.
Well, that'll be quite interesting. Just with regard, I think somebody might have asked this, but the circulation lifetime of these of the polymeric micelles, I think you did show it, but I couldn't quite get what the circulation lifetime actually is that you routinely observe. I mean, do you have half-lives of is it 10 hours or 2 hours? Or I wasn't quite clear on that.
Yeah, I think it is around 10 hours for this drug, if you're asking for the drug, but for the micelles themselves, it is longer than that, but with the drug. So, yeah, unfortunately, I don't have the data here, but I can share that with you. We calculated actually the pharmacokinetic parameters from different tissues, but I can share that later on. And
well, those graphs are informative anyway. No, I can kind of see this last.
Yeah, if you want to, yeah.
No, it's.
So these are just comparison of the AUC here, but we have actually calculated the pharmacokinetic parameters in different tissues. And I guess someone wanted to see the biodistribution profile again.
I also have a couple of more questions when you're ready for them.
Sure. Yeah, I guess like I just wanted to show the biodistribution profile because someone asked to see it again. I'm not sure if they were looking at this polymer or the later one that we looked at. Yeah, go ahead. I am ready. I can stop sharing.
Okay, no, that's fine. You can continue sharing. You were concerned we might have overlooked a question in the Q&A, and there was an earlier question, but I assumed it was the same question that Dr. Cullis asked. In case I'm wrong, I'll read it out nonetheless, and you can decide if it's been answered or not. So that question came from Riyad Al-Srani, who wrote, in the first project, the drug was released from the PBCL in less than 24 hours, yet you see the high biodistribution of the drug in the tumor after 48 hours. How would you explain this?
No, the drug was not released from the PBCL micelles. Like this is the release profile. So less than, like around maybe 40% is released at 24 hours from the PBCL. But from PCL, we have high drug release. And that's why that we did not use this polymer at the first try. But then later on, when it came to the NMIN project, we tried a longer molecular weight of the PCL. And then, we were able, to control the release when we use a longer chain of the polymer. Does that answer the question?
I'll see if the questioner responds to that, but in the meantime, I have another question from Francisco Pesine, who wrote, have you modeled the release kinetics?
No, we didn't do that.
Okay. Next question comes from Himanshu Bhatt, who wrote, can we prevent drug release from liposome in plasma? It was seen in the case of Doxil that it is releasing into plasma also. Is there any simple approach to prevent this drug release in plasma?
Well, some drug gets released eventually, and in plasma. So I guess for the case, I guess the important thing is that you should have enough drug staying with the nanoparticle when it reaches the tumor to have some activity. But I think it's still, perhaps Doxil is the gold standard for preventing drug release, in the plasma, even, after all of these different nano formulations that have come into market, still, I think no one can match as Doxil in that aspect. And different nanoparticles, they may work differently, as you can see with this PBCL, like we see this delayed accumulation in the tumor, and maybe it is working different from Doxil, but there might be some ways. We are looking into some new nano-delivery systems combining liposomes and polymers, but again, this is something that we just started, and I cannot comment if that will be successful.
Okay, we're coming towards the end of the hour. I don't know if there's any more questions, but
We've answered them all.
Hopefully.
Okay, Afsaneh, I'd like to thank you very much for a great lecture, and certainly there's an interesting variety of systems that you're presenting there, and it's a these are you never know what's going to work the best, that's for sure, and it's there's some fascinating data. So thank you very much.